Hi everyone, today we're going to be going over the phylum Porifera, otherwise known as sponges. So to get started, I just want to go over the basics in terms of classification and systematics of where the Porifera phylum lies on the evolutionary tree of animals and invertebrates in general. And so if we look at this diagram to the right, we have here Porifera at the top, showing it as one of the earliest evolving phylums of animals. And so this is going to come into some of the things we talk about in terms of their structure and how they lived and how they still live, and why they are so simple is because they were one of the first evolutionary experiments, if you will, of animal evolution, and they still exist, so they're not necessarily a failed experiment. They work, but they're very, very simple animals, and this is why they evolved very early on, and so they don't have very complex functions. And so then next video we'll go over Cnidaria, which is, as we can see, is still very early evolved phylum, but they're a tiny bit more complex, and so on as we go down the list here of different phyla of animals. However, today we're just going to be talking about Porifera, or sponges. And so Porifera, to give a little bit of context behind that name, because it sounds kind of weird for sponge, it just comes from porous, and that's why they're called Porifera. And if you think about sponges, that makes a lot of sense. They're super porous, and their pores have openings called osteums. So now jumping right into their anatomy and body structure, they have ostia and they have cells, which we'll get into more specifically what each cell type is called in a sponge on the next slide. But in general, their cells use flagella to drive in water into their internal skeletal cavity called the spicule and out the opening at the top, or their osculum. And this is to bring food and oxygen in and eliminate waste. And so they're filter feeders. In more detail, their body structure looks like this. As we can see, the ostia, or pore openings, are shown here to bring water through and into the spongicle, or central cavity, shown here. And then we have the osculum at the top, which is the opening where the water goes out, eliminating waste. And then we have different types of cells shown in the zoomed in figures to the right in this image. And what these are pinacocytes, labeled as epidermal cells, shown in the figure on top. And then at the bottom figure, there's a zoomed in image of a choanocyte. And these have the flagella that help with the propelling and filtering of the water. And then they have a mid-skeleton called a mesohyl, and this contains archaeocytes, which digest food, transport nutrients, and eventually develop into sex cells. And it also contains sclerocytes and spongocytes, which secrete the skeleton. And you might not be thinking that sponges have skeletons, but they do. And sometimes they're made up of an organic protein called spongin, and sometimes they're made up of spicules, which can be made up of calcium carbonate, in which case they're calcareous, or silicon dioxide, or quartz, in which case they're silicious. And a lot of times sponges have both spongin and spicules, and this helps support their structure. However, in terms of paleontology, we'll be focusing mostly on the spicules, and this is because these are what get preserved. Because spicules mineralize into calcite and silica, these hard parts are what's going to get preserved. And unfortunately, that makes it hard to decipher the fossil record of sponges sometimes, because sometimes it's just a bunch of spicules that aren't actually in their original sponge structure anymore, and they're just spread out in sand grains and whatnot. So that's unfortunate. But sometimes they're still congealed into their sponge structure, and the type of spicule matters in terms of interpretation of type of sponge. So if we look at the bottom figure here, now we can see types of sponge spicules. These include, but are not limited to, monaxon, triaxons, tetraxons, and polyaxons. And these more specifically can be called diactine, triactine, hexactine, tetractine, and polyactine respectively. And sometimes these sponge spicule types can take on different morphologies, but still within their spicule class. So for example, you might have a triaxon that has one longer ray than the other two rays, or a tetraxon with a similar type of longer ray than the others. So all of these slight nuances and the differences in the spicule morphology, as long as they're consistent within the sponge specimen you're looking at, can tell you a lot about what type of sponge you're looking at in terms of the broader class, as well as more specific and all the way down to the species. And so later, when we talk about different classes of sponges and their types of spicules, as well as their structures and other characteristics. We'll talk about these again. But before we get to that, we have to talk a little bit about the differences in simple versus complex sponges. Now, these aren't necessarily taxonomic groups of sponges, but these are categories of sponges based on complexity. What we have here are three groups: Ascon sponges, sycon sponges, and leucon sponges. Ascon sponges represent the simplest morphological structure a sponge could have. Ascon sponges have an osculum, ostia, a spongicle, and choanocytes, just like all of the main structures we already discussed. But their simplicity comes with their walls, which are thin, and their canals, which are short. As we can see with the sycon and leucon sponges, we can see major folding going on, especially in this circled area of the sycon sponge. And these folds are called incurrent canals. These canals help to propel water into and out of the sponge and help filter more water through the sponge and basically feed it more, give it more oxygen. It grows faster, it grows bigger, it gets more energy, and it's just from these folds which have a more complex design than the simple Ascon morphology. Lastly, we have leucon, which we can already see is pretty crazy complex, and with this we see that it not only has an osculum, but it has multiple oscula. It has multiple pore canals like all the types of sponges, but it also has multiple out openings, or its oscula, which none of the other types of sponges have. Additionally, it has this crazy squiggly structure going on, and what's happening there is it just has really thick walls with many complex channels, and all of these channels have special functions to help propel water in and out of the sponge to help it filter feed. However, in the case of leucons, sometimes they can have relatively small spongicles, or their internal cavity, because of their complex thick wall structure. And all of these things we've discussed so far are leading up to help us determine what kind of sponge we're looking at when we're looking at a fossilized sponge or fossilized fragments of sponges. So to go over the major classes of sponges, we have Hexactinellid or glass sponges, we have Calcarea, we have Demospongea, and we have, I just realized that says Scleractinia, which is a class of corals, not sponges. This is a lesson to you out there watching this thinking I know a lot of things. That's actually supposed to say Sclerospongia, and because I'm too lazy to change it right now, which I could do, but I'm not, I am going to tell you about it. So I'm telling you, I told you, consider yourself told. Sclerospongia. All right, moving on. I don't actually have a picture here of Sclerospongia. Well, I guess depending on how you class stromatoporoids, which could either be included in Sclerospongia or a distant extinct relative of Sclerospongia, or not even in the sponge phylum altogether. I don't know. We'll talk about why its taxonomy is so controversial later, but basically I have here a picture of stromatoporoids, and this is because these are what we're going to be focusing on. These are the most important similar sponge or maybe sponge to that class and are very helpful fossils in ways I'll explain later. And then lastly, another very taxonomically controversial group is the archaeocyathids. These include some of the most geographically widespread and short time-ranged index fossils that you'll ever see, and so they're really, really important. However, we're going to discuss their crazy controversy in terms of their classification and all of their characteristics at the end of the lecture, because they are not really technically confirmed to be part of Porifera, so they're kind of their own thing. And we'll talk about why paleontologists came to that conclusion when we talk about all of their other characteristics at the end of this video. But before that, let's get into each one of the classes we just listed for Porifera. We have the Hexactinellids, which, like we said, are glass sponges. So what does this mean? This just means they have siliceous spicules. We talked about spicules either being silica or calcite, and these sponges have silica spicules, which is the same composition as glass, which means that their nickname, glass sponges, is pretty apt. Also, their time range was from the Cambrian to the recent, or now, and then they have triaxon or hexactine spicules with 6 rays at 90 degrees from each other, and this is also called cubic symmetry. And so we can see here at the bottom this example of a glass sponge, very white and pristine, which is very different from the calcareous sponge fossils, which are going to be a bit more creamy colored. And so that's one way you can distinguish between siliceous and calcareous sponges without having to do any chemistry on them. So moving to the class Calcarea, we have the same time range, Cambrian to now, but we have calcareous spicules instead of siliceous spicules, and their spicules can be a range of shapes. They don't just have to be triaxon hexactines like the glass sponges do. They can be monaxon, triaxon, polyaxon, pretty much any shape you could want. And some even have calcareous skeletons without spicules. So they're very diverse. Now, before we move on, I do want to point out in the bottom right picture, we have this sponge picture here that I promised you we'd zoom in on so you can see all the spicules. And so we have this zoomed in square here showing their spicules and their shapes. And I highlighted a few of the star-like spicule structures in red here so you can kind of see them. And so if we go back and forth, you can kind of see where the spicules are kind of in a structure. However, most of them are broken up. This is an old fossil, so there's a lot of broken up spicules. Moving on to Demospongia. These also lived from the Cambrian to now, and they are the most common living group of sponges, and they live in both marine and freshwater environments. However, unfortunately, their fossil record isn't as complete as a lot of other siliceous and calcareous sponges because they make their spicules out of the organic spongin material we talked about earlier, and these won't preserve as often. However, in terms of type of spicule, they have monaxons and tetraxons most commonly, and sometimes they have really irregularly shaped knobby-like spicule structures called desmospicules, and this is more typical of organic spicules. You don't have the perfect crystallized shapes because they're organic material. And the last class we'll talk about is the Sclerospongia. Now I have it spelled right. This goes from the Cambrian to now, and I'll add to that possibly the Jurassic to now. And this is because some people think these stromatoporoids are part of the Sclerospongia and that these Sclerospongia stromatoporoid sponges started in the Cambrian, and some people think that the Sclerospongia class doesn't start until the Jurassic and that stromatoporoids are just a distant relative. And some people put this class and the stromatoporoids completely in Demospongia, and some people put the stromatoporoids in with the Cnidaria, Foraminifera, or Cyanobacteria. So this is just a really crazy taxonomically confusing situation for the sponges here. But we're going to try and just talk about stromatoporoids and explain how important they are and a little bit about their structure, and maybe that will at least a little bit help you in your class. Sorry, I can't be of more help in terms of the systematics of this group. So in terms of stromatoporoids, they have a time range from the Ordovician to the Cretaceous. So they are completely extinct, but they are really interesting fossils and have a very distinct morphology. This is why they're so hard to place. They have features similar to Porifera, but they lack spicules, which is why a lot of people put them in with Cnidaria. They also have this layered, wavy structure, which makes them similar to bacterial mats like cyanobacterial stromatolites, and they also have been thought to be similar to Foraminifera. So these have very distinctive features that just can't be placed very easily. But regardless of what they are, they were very important reef builders for a lot of the Paleozoic, namely in the Silurian and Devonian. As we can see in the picture below, we have this specimen where we can see these wavy layers shown in the black lines now, and these wavy layers are pointed out here as being called laminae, and then we have vertical lines within these layers shown on both these pictures, the sketch diagram as well as the real fossil picture, and these vertical sections in between the layers, or the laminae, are called pillars. And then we have these bumps on top because of the waviness of the stromatoporoid, and these bumps are mamelons. I think I'm saying that right. And then they have a star-like pattern of canals that go through the mamelon bumps, and these are shown in the left side of the left image, and these are called I should practice saying these things before I start these. Astroorhidae? Okay, we're gonna go with that. Astroorhidae. And as we can see, the last thing I mentioned is that they rarely contain spicules, which is one of the reasons they were so hard to place in terms of classification. Before we move on, I want to zoom in on this picture to the right so that we can see just the amount of detail that these laminae and pillars and canals and galleries, or the spaces in between the laminae and pillars, are. And so we can see in this image that all of the laminae and pillars are very fine, delicate structures that are very, very distinct. And so this is super cool to find, and you'll immediately know what it is if you find this kind of pattern in a rock you pick up. And like we said, the last group that we would talk about for this lecture is the archaeocyathids. However, we're going to talk a little bit about sponge reproduction, sponge ecology, and sponge evolution before we get to that. And so on this slide, we can see that in the figure to the right, we have this diagram showing sponges producing both egg cells and sperm cells. Because they can produce both of these, they can reproduce sexually with these egg and sperm cells, which eventually form into larvae, which will form a new sponge. However, they can also reproduce asexually through budding from pre-existing sponges. Additionally, sponges can regenerate complete animals from just broken up fragments of a pre-existing sponge. And this is very useful for sponges because a lot of times after really strong storms, if they're living in a really shallow marine environment, they can be broken up like crazy by the waves. And then they can later regenerate themselves to become become an entire animal again. Another really cool thing that sponges can do is to form into gemmules, which are just little armored nodules of food-laden archaeocytes that can basically protect the sponge in a stressful or nutrient-poor area or period in time until that sponge's environment becomes favorable again, and then it can revive itself, which is insane to me. I mean, could you imagine if we just like go into a pod when, you know, we don't have enough food available to us and then come back out when we do? So these guys are pretty awesome that they can do that. In terms of where they live, even though they can live in pretty much any aquatic environment on Earth today, the majority, or about 80% of them, live in shallow marine waters where there is substrate available for them to attach to. Also, they prefer areas with moderate to strong currents because this increases the water flow through their canals, and so they can filter feed easier. And another beneficial thing for sponges is that they have relatively few predators. Basically, certain types of sea turtles, fish, snails, sea stars, and chitons might munch on them from time to time, but the most thing they're useful for is for protection. Some types of worms, arthropods, fish, mollusks, and protozoans seek shelter in their protective spongicles. And so I put here to remind myself, funny shrimp anecdote. That's because the book that I'm reading that is helping me make these lectures, which is linked below if you want to look into it, they explain this in a really funny way, or I thought it was funny. But basically they mention that sometimes when shrimp go into the spongicles for protection, they stay in there so long and molt a bunch of times, because shrimp molt to grow, that they get so big by the time they try and go out, they can't fit out of the spongicle. And so they are stuck in there. And sometimes when pairs of shrimp get stuck in the spongicle, they can get fossilized like that. And these fossilized shrimp pairs that are trapped in this glass ball are actually a common wedding gift in some cultures, because apparently the concept of these two shrimp being trapped inside this ball is symbolic of marriage. I just thought that was hilarious. So anyway, moving on. Many sponges are important reef builders, and we'll talk about kind of the history of different sponges being dominant in reefs at different times in Earth history on the next slide. But the fossil record is a little fuzzy for sponges because, like we said, the spicules preserve, but sometimes they're just spicules. They're not actually attached to a sponge animal in a fossil. And so because the whole sponge doesn't always preserve, we have had to piece together a general pattern of sponge evolution without an entire picture. And so with this, we'll look now at the evolution of sponges. So if we look here at this timeline from the Mesozoic, Paleozoic, first we have the sponge classes all evolve. We have the Hexactinellid, the Calcarea, the Demospongea, and the Sclerospongia, maybe question mark, all evolved here in the early Cambrian. Then what we have is the extinction of the Archaeocyathids, which I know we haven't talked about, but I thought for the purpose of the evolutionary slide, I should include it on this timeline because the Archaeocyathids were really important reef builders, and nothing in the early to middle Cambrian was creating reefs other than Archaeocyathids. And so after their extinction, other types of reef builders or types of sponges that could reef build started to evolve. So then we have the sponges, especially demosponges, become dominant reef builders in the Ordovician, and then we have tabulate and rugose corals diversify and kind of get into the reef building business as well. And then we had sponges become less important reef builders because of coral evolution and corals taking over the reef building business, if you will. However, one of the groups that didn't become less important in reefs was stromatoporoids. Coral strome reefs were really common around this time. Then you had the Devonian extinction, which caused a decline in coral strome reefs, and Hexactinellids actually took over the reef building business, which, if you remember, are the glass sponges. And then you had Permian reefs, which were largely built by calcareous sphinctozoan sponges. Then you had the Permian extinction, which thankfully all of the major sponge groups did survive. However, during the Mesozoic, they began to decline in their dominance. And at the end of the Mesozoic, stromatoporoids went extinct at the KT extinction boundary. And during the Cenozoic, all the major sponge groups continued to decline in their abundance and dominance to the point that today they are only minor contributors to most reef habitats. However, they're still very common on Earth today. You just don't see them building huge reefs. Now, finally moving on to the Archaeocyathids. Archaeocyatha is a group of sponge-like animals with a very enigmatic taxonomy, which we've already discussed. And because of this, paleontologists have tried placing them in with corals, sponges, foraminifera, stony corals, peculiar sponges, and even algae. However, now paleontologists lean toward them being some sort of failed sponge-like evolutionary experiment. So that's what we're going to treat them as for this video. And these Archaeocyathids had a very short time range. They only lived from the early to middle Cambrian. Cambrian period, which is only the first period in the Paleozoic. However, they weren't this just measly unsuccessful experiment. They were incredibly geographically widespread to the point where they had global dominance in terms of reef building. And because of this widespread of archaeocyathans, they make wonderful index fossils. And so we can correlate Cambrian period sections across the globe with these fossils. However, their complete success during their lifetime makes it really confusing of why they came to an abrupt end in the middle Cambrian. And so this is a very confusing thing, and it's something that I think is really cool to look into. So if you want to look into archaeocyathans and trying to figure out what happened in the middle Cambrian that made them go extinct so abruptly, even though they were so happily dominating the world, that would be a really cool thing to go into. So just a thought. And then after their extinction, their role was filled by sponges and corals. To get into a little bit of their morphological characteristics, they had pretty simple cone-shaped structures shown here in this picture. They have a holdfast at the base to attach them to their substrate, and they have an opening at the top of their skeleton similar to a sponge's osculum. And also, like sponges, they were very porous. However, similarly to cnidarians or corals, which we'll talk about next time, they had septa, which were vertical plates equally spaced throughout the space in between their two walls in their structure. And this space is called their intervallum. Additionally, they had horizontal plates called tabula, or plural tabulae, and bubble-shaped structures also in their intervallum called disepiments. And these are all characteristics shared with tabulate and rugose corals, and we'll talk about those in the cnidarian video. But these similarities to sponges and cnidarians both made them so hard to place, and is why we consider them a whole different group. In terms of ecology, the archaeocyathans preferred shallow marine carbonate shelves, and this makes sense if they were reef builders, and they were not really found in brackish or hypersaline water or deep water deposits. And they were also always associated with algal fossils when they're found in the rock record. And so this suggests some sort of symbiotic relationship between the archaeocyathans and algae. And this is analogous to hermotypic corals today. Hermotypic corals are corals that have symbiotic relationships with algae called zooxanthellae in their polyps. And these algae can photosynthesize and help the coral to get energy and food to grow. And this may have been a similar relationship between algae and archaeocyathans in the Cambrian. And this may have been the first time that that kind of relationship evolved, but we don't know for sure. This is speculation based on what they're found with in the fossil record. However, what it does tell us for certain is that the archaeocyathans like to live in the photic zone because the algae fossils they're found with also needed to live in the photic zone to get sunlight. Additionally, their simple conical structures were ideal for filter feeding, just like sponges and corals, through their pores and out their osculum. This beneficial conical structure for filter feeding explains their success in the early Cambrian, but it doesn't explain their abrupt extinction. And like I said, this remains a mystery. So I hope that you find that as cool as I do, and I hope that you're excited by the fact that we don't know every single thing about different phyla and their evolution throughout Earth's history. And I hope it excites you to get out there and figure out things you're curious about. Go study things you're curious about. It makes your life a heck of a lot of fun to just work on the things you love. So go for it. If you find this interesting, I encourage you to go solve the mystery of archaeocyathans and what they are and why they happened. And when you do, let me know what the answer is. I'd be very curious to hear. So anyway, that is all I have for you guys. I hope this was helpful. And if you want to check out the rest of the paleontology videos, you can click the rectangle down below to go see the entire paleontology playlist. Anyway, thanks again for watching. I'll see you guys next time. Bye.